Good afternoon, everyone. My name is Yusminata Pilatu. I'm a senior research scientist at the Center for Deep Sea Research, Indonesian Institute of Science, LIPI. And today I'm going to present to you my paper entitled Ice-Dispersed Ketosana Nanoparticles Effectiveness in Fresh Russell's Mackerel Scat Decapterus russelli Preservation. The title is a bit modified due to grammatical purpose. So why this research? Well, to start with, Decapterus russelli is one of the species of the fish included in the Decapterus genus. And it's included in a major commodity in the Ambon traditional market. Ambon Municipality Statistical Bureau recorded in 2019 that there were 5,122 tons of the captured species that were auctioned in the local fish trade. Now, globally speaking, this genus, the Decapterus genus, is also important in terms of fish market. Why is that? Because the Decapterus species has better flavor and affordable in terms of price, thus preferred by locals. Now, the local fishmongers need an alternative preservation method that is not only low cost, but it is also non-toxic. And ketosana nanoparticles has these two features, among other features that it has, like the most important one is that is safe for human consumption and that is biodegradable and it can be generated from seafood waste such as shrimp shells. So we already done previously a study regarding the effectiveness of nanoketosan in maintaining a fresh yellowfin tuna um, freshness. However, there is a gap in this research area in the sense that we are not we are not sure yet about the possibility of ice dispersed ketosana nanoparticles in maintaining small pelagic fishes, especially during marketing hours. So the study's objectives are first to assess the ice-dispersed ketosana nanoparticles effectiveness in fresh catfish preservation compared to plain ice. And the second is to assess the ice-dispersed ketosana nanoparticle effectiveness when made with still water compared to seawater. The importance of this study is, first of all, to provide information regarding innovative preservation method exploration for local fishmongers in maintaining the saleability of their products. And the second one, we would also like to know about the seawater quality in the local fish market because often the local fishmongers use this seawater to freshen the fish that they sell. So to do this experiment, first of all, we purchased the Russell's mackerel scat from the landing site. And once we purchased them, we transferred them directly in a cool box at a temperature of 14 degrees to the laboratory and where we weighed all the fish and we incubated them with the ice tubes prepared previously before the experiment, like 2 days prior. And the ice tubes were made from two types of water, which were seawater and distilled water. And inside which we add or we didn't add the ketosana nanoparticles. And during the incubation period, every 3 hours, we took samples to do the total volatile base, trimethylamine, total plate count, analysis and also to measure the pH level. And we also did the organoleptic assessment in parallel. So the results that we obtained, first of all, for the total volatile base, we can see here that the fish incubated with tube ice dispersed ketosal nanoparticles made with distilled water was still below the rejection level, which is shown here by the dashed red lines at 30 milligrams nitrogen per 100 gram samples, and that until 12 hours of incubation, um, the samples, the fish, were still below 30 milligrams, and it's at the 12 hours that it reached 30 milligrams, contrary to the other three treatment, in the sense that the other three already reached 30 milligrams at the 9th hours of incubation. Now, this result was similar with what we observed in the 3-methylamine analysis, in the sense that the fish incubated with ketosal nanoparticles added in distilled water to make ice, it was the lowest compared to the other three treatments. Now, we also here try to see the relationship between the TMA index and odor score, and what we found here is that the TMA index negatively correlated with odor score, which makes sense because the rejection level was below 7 for the odor score, and this relation is statistically significant at a 95% confidence level. Concerning the total plate count, the result was actually in line with the total volatile base, in the sense that even until the 12 hours of incubation, the total bacterial count was below the other three treatments. So here the rejection level, which was shown with the red dot lines, was at the logarithmic scale of 1 times 10 power 5, or 5.69, the value. And however, for the pH level, we don't observe too many difference, in the sense that they all the four treatments follow the same tendency, which were decreasing below 6, and this could be due not only to the breaking down of amino acids in the fish meat, but also could also due to the ingredients of the ketosal nanoparticles solution. And concerning the organoleptic assessment profile, what we can observe here is that the profile of the samples were similar between those that were incubated with tube ice made from distilled water, added or without ketosal nanoparticles, compared to those that were prepared using the seawater. And this is interesting because here apparently even the ketosal nanoparticles could not prevent the growth of bacteria already present in the seawater. So to conclude, what we can say here is that the tube ice dispersed ketosal nanoparticle could prolong catfish freshness up to 12 hours or 3 hours longer than those without. And second, the ice made with distilled water were more effective in maintaining catfish freshness than those made with the seawater in the fish market location, which indicated the low quality of seawater. So I would like to end my presentation by acknowledging Tamara Ginsel, who was the co-author of this work, but I also want to thank the UGMT, which has contributed generously the material used in this project.